This is the second video for Unit 17. It includes Part E, how does the immune system do it? Part F, how are specific T cells and antibodies produced? And Part G, how are antigens neutralized or destroyed? How does the immune system do what it does? There are many killing agents involved in the immune response, agents that actually attack and destroy the antigen. There are cells, we call them cellular effectors. They attack the antigen directly. They include killer T cells, also called cytotoxic T cells, macrophages. Both of these types of cells are very important. Then there are neutrophils and eosinophils and others. More is being learned all the time. There are molecular effectors. They include complement, cytokines, and others. Notice we have not listed antibodies among these agents. Why? Because they fall into the category of other necessary agents. These include memory cells, B and T cells. They include B cells that mature into plasma cells and secrete antibodies. These agents, although they don't directly attack the antigen, are critical for the identification and destruction of antigens. How does the immune system know what to attack? Cellular effectors don't simply wander around aimlessly. The immune system must be able to identify the antigen, and it uses what I call recognition proteins. For every kind of antigen, there is a different recognition protein. Recognition proteins are complementary to the antigen. They bind to the antigen. They include T cell receptors and antibodies. Both are roughly Y-shaped molecules. T cell receptors are produced by and attached to the T cells. Antibodies are produced by and usually secreted from the B cells. Specific immunity takes two forms. One is called humoral, or antibody-mediated immunity. This uses the B cells. Now, why is it called that? Well, body fluids once were called humors. Humoral immunity was called that because it was first found that body fluids, especially plasma, contained something that contributed to immunity. Later, the antibodies were discovered. Cell-mediated immunity is immunity using the T cells. Humoral immunity involves B cells, a type of lymphocyte, including plasma cells, which are mature B cells that produce the antibodies. Antibodies function best against antigens on bacteria and free molecules. Cell-mediated immunity, on the other hand, involves T cells. This is another type of lymphocyte. It includes killer T cells, helper T cells, and other types that are being discovered. T cells function best against antigens on eukaryotic cells, such as parasites, tumor cells, transplants, and virus-infected cells. In humoral immunity, the antibodies produced by the B cells are secreted by the B cells or attached to the plasma membrane to serve as receptors. Let's look at the structure of an antibody. Antibodies are a bit more complex than the simple Y shape that we've described earlier. These are globular proteins. They contain a carbohydrate part, so technically these are glycoproteins. They're produced by the immune system, usually in response to an antigen. You remember the anti-RH antibodies. They were produced when the person was exposed to the RH antigen. Some antibodies are permanent. You remember those in the ABO blood type. Those were permanent antibodies. They're located in the gamma globulin fraction of the blood, and they're called immunoglobins, capital I, little g, because they're globular proteins involved in immunity. They comprise roughly 20% of the circulating plasma proteins. They're Y-shaped, as we've said, and they're composed of 4 polypeptides. 2 of them are heavy chains, and 2 are light chains. A variable region has many, many forms to bind with millions of different antigens. A constant region exhibits fewer forms, but it has an effector binding site. The antigen binding site, referred to as the valence, is found at the tips of the variable region. Bivalent antibody has 2 binding sites. A multivalent antibody has many binding sites. The effector binding site, as we mentioned, is in the constant region, and it attaches to cells and molecules of the immune system. Antibodies function by 2 actions. One is by direct action against the antigen. They do this to prevent spread of antigens, to prevent attachments of antigens, but more importantly, they function in an indirect way against antigens by mediating other functions such as attracting phagocytes, releasing phagocytosis, activating or fixing complement, promoting inflammation. Let's look at the classes of antibodies. This table is divided into 5 columns. The first column lists the immunoglobin alphabetically. The second column lists the percent in the blood. Now, this is when no infection is present. The percentage in the blood will change when there is an infection. Third column shows the structure, and the fourth, the binding site. And then the last column is a very general function, which we'll give a little more detail of in the next slide. Immunoglobin A occurs in 2 forms, a secretory form and a serum form. The serum form is not well understood, but the secretory form makes up about 15% in the blood. It consists of 2 of the Y-shaped molecules, and so it would have 4 binding sites. It's found in secretions, and so doing it protects mucosal surfaces. Immunoglobin D, not very common, less than 1%. It consists of a single Y-shaped molecule with 2 binding sites, and it serves as receptors on the B cells. Immunoglobin E, likewise not very common, consists of a single Y-shaped structure with 2 binding sites. It stimulates basophils and mast cells to secrete histamine and other active substances. It also functions against certain parasites. Immunoglobin G makes up 75% in the blood. This is the one that we usually refer to when we're talking about typical immunoglobin activity. It's a single Y-shaped molecule with 2 binding sites. It functions most importantly in the secondary response. It increases phagocytosis and fixes complement. We've mentioned complement several times already. We'll come back to its specific function at a later time. Then there's immunoglobin M. It makes up about 10% of the antibodies in the circulation. It consists of 5 of the Y-shaped molecules. Therefore, it has 10 binding sites. It tends to function more in the primary response. It increases phagocytosis and fixes complement. Let's look at a few other details. Immunoglobin E, the secretory form, it's found in secretions, as we said. They include milk, tears, saliva, and mucus. They prevent viral and bacterial attachment to mucosal surfaces. Their numbers tend to decrease in stress. One reason people seem to get sick easier during stressful times. Immunoglobin D is a receptor on the surface of the B cells. Immunoglobin E is a receptor on mast cells and basophils. It's present in the mucus of the nose, throat, lungs, and endothelium. So it protects internal body surfaces. It's involved in allergic reactions. Immunoglobin G. It's a stable, long-lasting antibody. It provides naturally acquired active immunity. It can cross the placenta because it's a smaller molecule, and so it provides some immunity to the fetus before birth. Immunoglobin M has a short lifespan, but it's very efficient because of the 10 binding sites. It includes the ABO antibodies. What about cell-mediated immunity? T cells have T cell receptors. These are similar to antibodies with an effector binding site that attaches the T cell receptor to the T cell. They also have antigen binding sites that attach the T cell receptor to an antigen. And in so doing, it finds specific germs to attack. T cells destroy the antigens. They first of all find the foreign cell. They bind to the target cell, the foreign cell. They inject the cytotoxic substance and kill the foreign cell. So they're involved in direct attacks. Cell-mediated and humoral immunity have several main points. Millions of different T cells exist. Each has its own T cell receptor, so there are millions of different T cell receptors. A different T cell receptor exists to match every possible antigen shape. PCRs bind specifically to the antigen that activated the T cell. Millions of different B cells exist. Each produces its own kind of antibody, so there are millions of different antibodies. A different antibody exists to match every possible antigen shape. Antibodies bind specifically to the antigen that activated the B cells. Notice the specificity we're referring to. PCLs bind to specific antigens. Antibodies bind to specific antigens. These are the ones that the immune system intends to attack and destroy. Antibodies and T cell receptors also attach to cells and molecules of the immune system. Now notice what we're saying. They can attach to the antigen, but they also attach to the cells and molecules of the immune system, and so they serve as links between the two. Let's look at the key step. Number 1, the antigen enters the body. Number 2, specific T cells and B cells complementary to the antigen are activated. The activated T cells have T cell receptors that match the antigen shape, that is, they're complementary. The activated B cells secrete antibodies that are also complementary to the antigen. Step 3, the T cells and the antibodies find the antigen, and the T cells and antibodies help to destroy the antigen. The details are covered in the next sections and include how specific T cells and antibodies are produced, how the antigens are neutralized or destroyed. Before we start, let me introduce you to a few terms. Since antibodies are the best indicator of the presence of an antigen, the terms involved in the immune response generally refer to antibodies. An antitoxin, for example, is an antibody against toxins produced commercially. Antiserum refers to a serum containing antibodies. Seropositive refers to antibodies against a particular antigen or present. Seroconversion is the formation of antibodies against a specific antigen. How about control of the immune response? Since the immune response is a very, very destructive system, there has to be a way to turn down the response. Failure to do so results in various autoimmune diseases. and so your immune system begins to attack your own tissues. Control is accomplished in many ways. They're not well understood. For example, elimination of overactive T cells in the thymus. This is called central tolerance. A type of T cell called the regulatory T cell. This was discovered in 1995 and led to a Nobel Peace Prize. This is involved in peripheral tolerance. So, part F, how are specific T cells and antibodies produced? Let's look at some of the effector cells and molecules involved in the immune response. There are molecular effectors. They include specific receptors. These are complementary to the antigen. MHC molecules. Think of these as a holder for the specific receptor. Antibodies will be illustrated by the Y, and signal molecules. The antigen will be shown as the heavy red arrow. Cellular effectors include antigen-presenting cells, APC, and helper T cells. They also include the B cells and the T cells. Once ingested by the antigen-presenting cell, the antigen is processed. Now, this may be complex, but essentially it means it's cut in pieces. Then step 4, a fragment of the antigen, this is called an epitope, is inserted on the surface of the antigen-presenting cell. It's held in place by an MHC class II molecule. This serves kind of as a holder. This is called antigen-presenting. The MHC class I molecules, which are also found in the immune system, are involved more in processing non-foreign antigens, such as tumor antigens. Antigen transport then occurs. Step 5, the antigen is transported by the APC to secondary lymph organs, from the skin to lymph nodes because they're nearby. These are transported by the lymph vessels, from mucous membranes of the nose and mouth to the tonsils because they're not far away, from the mucous membranes of the intestines to the Peyer's patches. These are like lymph nodes, but they're in the wall of the intestines. Specialized cells do the transport. And from the skin, mucous membrane, and other sites to the spleen by the blood. So you see, these secondary lymph organs are where the antigens are being transported for further processing. T and B cells, meanwhile, are circulating among the primary lymphoid organs, the thymus and the patches in the bone marrow, to the secondary lymphoid organs. They're going there to look for antigen-presenting cells with antigens. Step 7. In the secondary lymph organs, the antigen-presenting cell finds helper T cells, which have a receptor complementary to the antigen and activates the helper T cell. These helper T cells are also called CD4 T cells. So imagine what's going on. The antigen-presenting cell wandered into the secondary lymph organ and searched until it found the right helper T cell. Step 8. In the secondary lymph organs, the helper T cell sends signals. These signals will activate complementary B and T cells. Now again, you have hundreds of thousands of mixed B and T cells. The helper T cell is trying to find one of each that has the same kind of, that is a similar receptor. The signals that are sent vary. There are many different ones, but interleukin-2 is an important signal. Remember, as I said, millions of different T cells exist. Each has its own kind of T cell receptor, so there are millions of different T cell receptors. Likewise, millions of different B cells exist, and each produces its own kind of antibody, so there are millions of different antibodies. In the secondary lymphoid organs, the activated B cells and T cells begin to multiply. This is called clonal selection. The B cells mature into plasma cells and secrete antibodies. Millions of antibodies are secreted. The antibodies are transported by the blood and lymph to the site of infection. Meanwhile, the T cells mature into killer T cells, cytotoxic T cells. They're also called CD8 T cells. The killer T cells are transported by the blood and lymph also to the site of the infection. So you see, we're mobilizing the defenses to fight off the infection. In the secondary response, this is a subsequent exposure to the same antigen. So what we just described would happen either in a first or secondary response. The secondary response requires long-lived memory cells located in the secondary lymph organs. The memory cells are probably numerous B cells and T cells that are complementary to the antigen, and they're primed to multiply very fast. So the immune system, in effect, remembers the original antigen when exposed to it again. It stimulates a rapid production of killer T cells and B cells. Okay, we have seen how antibodies and killer T cells are produced. Now let's look at what they do to neutralize or destroy the antigen. B cells have multiplied and produced antibodies complementary to the antigen. T cells have multiplied and created killer T cells, cytotoxic T cells, complementary to the antigen. Remember the structure. The antibody has an antigen binding site and an effector binding site. The effectors include macrophages, complement. T cell receptors bind to T cells with the effector binding site. They bind to the antigen with the antigen binding site. The antigens may include parasites, cancer cells, transplanted tissue. The antibodies act as a link between the effectors and the antigens. Let's use macrophages as an example, but many other cells of the immune system would also do this. The macrophage contains binding sites for the effector region of the antibody. The antibodies will find specific germs to attack by forming attachments between the antigen binding site and the antigen on the germ. And remember, the antibodies have been produced specific for that particular agent, germ. They form another attachment between the effector binding site and the macrophage. Let's look how it works, and we'll compare the efficiency of the actions with and without the antibodies. Without antibodies and with antibodies, this will be a chart. The circle represents the antigens, the germs. The Y is the antibody, and the C with a square around it represents complements. Let's take, for example, phagocytosis, which is a very important function of the immune system. Macrophages and neutrophils carry out phagocytosis. Without the antibodies, they wander around aimlessly, maybe accidentally coming across a germ, an antigen, and destroying it. But with the antibodies, they know exactly what to do. They attack the antigens, the germs. Phagocytes are attracted with complement C5a, with kinins, and with other substances, and it works something like this. The antibody will bind to the antigen by the antigen binding site. The antibody will bind to the macrophage by the effector binding site. And so the antibody acts as a glue, a link between the two. Macrophages have receptors for antibodies. Mast cell granules contain antimicrobial chemical mediators and vasoactive substances to increase inflammation. Without the antibodies, they fail to release the substances. With antibodies, they release the substances and making the immune system much more effective. The mechanism: antibodies bind to antigen by the antigen binding site. The antibodies bind to mast cells by the effector binding site. The mast cells have receptors for the antibodies. Microorganisms and molecular antigens, toxins, increase the infection by spreading. Without antibodies, they spread easily. With antibodies, they cannot spread. Why? Because the antibody binds to the microorganisms by the effector binding site to prevent their spread by agglutination. They form immune complexes of molecules or agglutination of cells. This is just what happened when we described the agglutination of red blood cells in a mismatched transfusion. In order to infect, the microorganism has to stick to a surface: skin, mucous membranes, cell membranes, and so forth. Attachment of the microorganism to the surfaces is a common form of infection. For example, Neisseria gonorrhea, the causative agent of gonorrhea, has to attach to mucosal surfaces of the reproductive tract. Without antibodies, they stick. With antibodies, they cannot stick because the antibody has bound to the microorganism and blocks its attachment, blocks its binding site. Antibodies bind the complementary C3A by the effector binding site to activate the complement system. Now hang in there. I know I keep referring to complement and I haven't talked about it yet, but it's coming up. Complement system will be described. Antigens are killed with killer T cells, bind to the cell surface antigen that activated them, inject cytokines, some of them are called perforins or granulolysin, into the foreign cell to kill it. Eosinophils, for example, do this. They release hydrolyzing or digestive enzymes. Complement C9 does this. So, what is this complement system we keep talking about? Well, it's a complex system of proteins. Many of them are called complement proteins. Some are called properdin proteins that increase the efficiency of the immune system. They were named complement 1, complement 2, that is C1, C2, C3, hopefully in the order that they act, but it turns out some of them are out of order as they learn more about them. We have simplified the complement system to represent only 3 of the complement proteins. It has 2 pathways. One starts with antigen antibody complexes. It's called the classical pathway. And then there's the alternate pathway that begins with cell walls of microorganisms, particularly bacteria. To fix complement is to activate the complement system. Let's look at the classical pathway. It begins when antibodies bind to antigens, so this depends on the antibodies. The antigens are mainly bacteria that become coated with antibodies or antibody-antigen complexes. These are the immune complexes, and it's very specific. It activates C3. The alternate pathway begins when bacterial cell walls bind to properdins, and these are not dependent on antibodies, so it's non-specific. They activate also complement 3. What happens? Well, C3 splits into C3A and C3B. C3A increases inflammation by increasing the release of inflammatory substances from basophils and mast cells. It increases neutrophil and macrophage activity. So you see, it does a lot to increase the process of inflammation. C3b, on the other hand, increases phagocytosis. It also binds to antibody-antigen complexes, binds them to red blood cells, and they play a role in destroying antigens because the liver will then take up those complexes and destroy them. And this is called immune clearance. C5 becomes C5a, and this serves to attract phagocytic cells. C9, finally the last one activated, this actually causes cell destruction, cell lysis, by punching holes in the cell membrane. It forms a membrane attack complex from complement proteins. Here you have 5 of them, and they actually punch a hole in the membrane, and the cell will rupture because the contents immediately leak out. And that is a whirlwind look at the immune system. You're now ready to progress to Unit 18, Circulatory Anatomy.